application. So plant tissue culture is a method by which plant cells can be cultured artificially in order to grow plants or plant organs. So this is an overview. Before we move on to plant tissue culture technique, we should understand three fundamental abilities of a plant cell which makes it possible to culture in vitro. First capability is known as totipotency. Totipotency simply means the potential of a plant cell to give rise to an entire plant. And this is kind of like a totipotent stem cell, which can give rise to an entire organism. So, any cell from plant can be cultured and can be ultimately induced to form a mature plantling. Second capability is known as dedifferentiation and redifferentiation. Differentiation simply means specific lineage commitment. So once a plant cell is committed to a particular tissue type or cell type, that is known as differentiation. And these differentiated tissues can be de-differentiated. That means once specification is done, the specification can be reversed and it can get back to a undifferentiated mass, which can be further redifferentiated to a completely different substance. So this phenomena of differentiation, de-differentiation and redifferentiation makes it possible to culture plants, and several aspects of tissue culture is possible due to this phenomena. And third option is competency, which simply means the endogenous potential of a given cell or tissue to develop to a particular way. So it's kind of like a genetic program which is inbuilt and which is helping or guiding the cells to grow into a particular fate. So these three factors are absolutely essential for plant tissue culture. Let us talk about the advantage and disadvantage of plant tissue culture in a moment. So, first of all, the new plantlets can be grown in a very short amount of time. So you can bypass the long time to produce a plant in a field. Only small amount of plant material is required. A small portion of the leaf or a small portion of the meristem is more than sufficient to grow an entire plant. So that's the biggest advantage of this technique. New plantlets or plants are more likely to be free of viruses or any kind of diseases. And this is really important for production of virus-free or pathogen-free plants. The process is not dependent upon any season, right? So you can enjoy, let's say, orange even in scorching summer. So this is the biggest advantage from an economic point of view. And people who are looking to cultivate challenging plants which are not grown in a particular region, if they want to grow them, the soil conditions or the atmospheric conditions might not be favorable for that. So a laboratory setup or a in vitro situation might be advantageous for that fact, right? But there are certain disadvantages associated with these tissue culture system. First, tissue culture can require high labor and it could be very costly, at least the initial setup. For many of the agriculture dependent countries, which are third world countries, it is absolutely difficult to invest so much of money on the initial setup. Now there could be chance of plants to be uh, less compatible with the environmental conditions because they are growing in a sterile environment and they are less resistant to diseases or less resistance to environmental changes. So there could be a big question mark whether these kind of plant tissue culture techniques can be appropriate for every environmental situation. And lastly, there could be failure of growth of plants. There could be aberrant effects in the plant tissue cultures which leads to disease plant or imperfect growth. So these are all the disadvantages that one should consider while thinking of performing a tissue culture experiment. Now let's talk about the factors that affect plant tissue culture. First of all, the growth media, because we are growing the plants in vitro. So that is why we have to ensure that proper minerals, growth factors, carbon sources, and hormones are present in this growth media which can support the plant growth. After that, temperature, humidity, sunlight, all of these conditions has to be met for a proper plant growth, and that need to be provided artificially. Genetic factors are always present for a proper growth of plant from a tissue culture setup. Lastly, the explant source, the starting material we are using for growing a plant. Obviously, a tissue which is less differentiated has a greater chance, or it would be best for making a new plant compared to a tissue which is totally differentiated. Now let's talk about the basic components of the plant culture media. It should have all the essential elements, the macro and the micronutrients that are important for plant growth. It should have plant hormones such as auxin and cytokinin and other hormones which really helps in plant growth. It should have a carbon source. Generally, the carbon source is actually sucrose. And ultimately, it should have a supply of organic elements such as vitamins and amino acids, which are important for anabolic responses in a plant. So at least these four group of factors are essential for a plant to grow in a tissue culture setting. Now let's talk about the tissue culture types. So at least there are these 6 tissue culture types, including whole embryo culture, seedling culture, organ culture, callus culture, suspension culture, and protoplast culture. So each of these culture methodologies has their own advantage and disadvantages, and they can be utilized in particular purposes. So in subsequent time, we would look at these particular applications. First, let's start with callus culture. So callus formation in plant can be induced in several ways. Callus simply means a growing mass which is unorganized and undifferentiated, and these are basically parenchyma cells. Now, plant callus can be derived from somatic tissues. Callus formation is induced from a plant tissue sample after surface sterilization, and it is important because callus can be infected by other pathogens or it can be contaminated. So that is why maintaining a sterile environment while performing a callus culture is absolutely essential. The cells that give rise to callus or somatic embryos usually undergo rapid division or partially undifferentiated, such as they could be also partially undifferentiated, such as meristem tissues. Now, there could be the explant source, and while choosing the explant source, we have to ensure that we are choosing a good explant source that can give rise to a plant. For example, if we compare between a leaf explant and a meristem explant would be far better option for growing a callus. Now, placing that explant into a particular medium such as MS medium or Murashige's Cook medium is the first step of callus formation. After putting it into the medium and waiting for several days, the callus would be formed, and it looks like these tumor-like tissues which are kind of undifferentiated. It doesn't have any shape or size, and when it is um, taken into account from a molecular point of view, they are undifferentiated. Their lineages are not committed. So these callus would be eventually maturing into a particular somatic plantlet, and that can be placed into a soil or they can be grown artificially. So from these callus, an entire plantlet can be formed.